Hello, this is going to be the last video of the electron transport chain and ATP synthesis, and we're going to see, we're going to discuss some subtles that are moving in and out of the mitochondria, certain things. The problem is, again, that the inner mitochondrial membrane is so impenetrable to anything. Even protons cannot go through. That's the whole point of the electron transport chain and building the proton gradient that you need across the membrane so you can drive ATP synthesis. So how are things going to go through? It's if it's so super tight. It's nice to be super tight because that's when you have a really efficient battery, but that generates other problems. So the cell has to solve these problems. Um, this is one obvious problem is, you made all the ATP, which is made in the matrix by the soluble part of ATP synthase. Well, you really need it, outside of the inner mitochondrial matrix. So you have to get it out. ATP is supercharged. Of course, it has all these negative charges. How is it going to come out? So luckily, there is a carrier protein that swaps ADP with ATP. For every ADP that comes in, an ATP goes out. So that's convenient. That's very nice. Um, and this is all fine. You have a lot more ATP. This is thermodynamically favorable. You have a lot more ATP on the inside, so it wants to go out. You have more ADP on the outside, it wants to come in. So that's all perfect. It does cost something. However, when you do this, you do lose a proton from the outside. Okay, so it does involve some minor proton loss, which of course now that you are thinking in electric terms here with a battery, it will decharge a little bit. You're going to lose some of that charge, when you move the ATP back out. But it is what it is. Um, a bigger problem is how do you bring the NADH from glycolysis into the mitochondrial matrix? Remember, the NADH from the TCA cycle, it's already in the matrix, so you don't need to worry about it. But if you do glycolysis and then pyruvate dehydrogenase from pyruvate and go all the way to the TCA cycle and so on and so forth, you have made some NADH in glycolysis. So what's going to happen to that? How is that going to be, or can it even be used to enter the electron transport chain and eventually contribute to the charging of the battery and making of ATP? The answer is yes, but you have to bring it in again. And so it needs help. So you need shuttles. So there are at least a couple shuttles for NADH. One is the glycerophosphate shuttle. And what happens in this case, you have glycerophosphate shuttle. So what it does, it takes NADH and it reduces. You have the redox state of the glycerol here, okay, which is glycerophosphate. You go from this hydroxyl group, actually, you go from this keto group to a reduction. You make glycerol in this case, right? So you use a reducing power of an ADH to get the keto group and make it a hydroxy group, and then do the converse redox here using FADH. So with complex 2. So that can be used again through complex 2. Remember now what the problem is. And once you are in complex 2, then you can reduce Q, of course, and then you go on to complex 3, 4, and so on, as we have discussed. But the key point here is the NADH from glycolysis. Now, this is all the super nice NADH is converted to FADH. So you bypass complex 1. Remember, complex 2 does not translocate protons. Okay. If you had found a way to feed an ADH to complex 1 instead, then you could get a few extra protons coming out. But now if you go through this route here, you lose those protons coming out, right? So it misses complex 1, you have less proton transport and less ATP will be produced. Now, luckily, this is a minor subtle, okay? Um, the majority of NADH from glycolysis goes through a different cell, which we're going to see a few times in this course. So we're not going to describe all of it. It seems a little complicated. So we're just going to focus on one or two reactions here, and that's the malate aspartate cell. So what you do is you do this reaction that we had seen the TCA cycle. In the TCA cycle, you go from malate to oxaloacetate. That's the last reaction that closes the TCA cycle. But outside in the cytoplasm, you go in reverse. You go from oxaloacetate to malate, which is actually a very favorable reaction that we had discussed all these delta E values. We had calculated it. So it wants to go from in the cytoplasm. It wants to go from the oxaloacetate to malate, and in the process, of course, you use NADH. So imagine now NADH that you use in the cytosol. Remember, these NADH can go through the outer mitochondrial membrane into in the intermembrane space. That's no problem. It's pretty open to transport. This NADH is used, so glycolysis here brings in NADH. You use it to reduce oxaloacetate to malate. Malate then gets picked up by a specific transporter, which brings it in the matrix. And at the same time, the same transporter brings out alpha ketoglutarate. We're going to see what happens to alpha ketoglutarate, how everything gets equalized in the end. But once now malate is in the matrix, it's just a normal reaction of the TCA cycle. You go from malate to oxaloacetate, and we saw how now in the matrix this is possible. Okay, and you make NADH. This is possible because then it's coupled to citrate synthase and so on and so forth. So it drives it forward. But the net result for what we're talking about is you took NADH from the outside and you brought it inside. And now that NADH can go through complex 1 and translocate all the protons and so on and so forth. So those are two ways, these shuttles, that you can bring in NADH. And this is more efficient. You get a full complement of ADP produced because you do not miss complex 1. There's something interesting that just came out actually very recently, again in a very prominent journal. So many cancer cells have a mitochondrial dysfunction. They are defective in their electron transport chain. And what happens in this case, they do something really sneaky. They bring in glutamine, and from glutamine, as we will learn, later in the class, you can make alpha ketoglutarate, and in a sense, you go backwards, anti-clockwise, on the TCA cycle, and from alpha ketoglutarate, you make citrate. Okay, and then through the subtle that we discussed, you can get oxaloacetate and malate, right? And but this malate doesn't go in. Okay, so instead, okay, you generate NAD plus. This is the same reaction. Let me go back in a second. This is this reaction here. Okay, and when you go from oxaloacetate to malate, you generate NAD plus. Okay, you have oxidized NADH now. And why is that important in the sneaky cancer cells? Because this NADH plus can be used in glycolysis. Remember, this is extremely important because the point of fermentation that we had discussed earlier, so the glycolysis can run all the time and make ATP, let's say in cancer cells, is the point of glycolysis of fermentation was to regenerate the NAD plus. And but in this system, okay, going through glutamine and making oxaloacetate eventually that can participate in this malate aspartate shuttle here, you can in effect regenerate NAD plus. Okay, and that will allow you to keep running glycolysis and make ATP from glycolysis on the face of mitochondrial dysfunction. So this is another way that cancer cells can keep satisfying their biosynthetic needs, okay, even if they don't have, functional electron transport chain. They cannot do oxidative phosphorylation. They like to do a lot of glycolysis. They get all the biosynthetic intermediates from it. They run many of the TCA cycle reactions. They just don't do the electron transport chain here. And they don't care about making too much ATP as long as they can make enough to keep going. And all the biosynthetic intermediates, they're all set because then they can build new cells, essentially. All right, so that's the involvement of this shuttle in cancer